So today's topic is genetic uh, frontiers, and we will talk about the genetic modification of the organisms. And you probably heard about genetically modified organisms in the food because it's very common uh, to label the food that it does not contain genetically modified organisms or GMO. So it's the same in Russian is GMO, uh, генно-модифицированный organism, and and recently it was like a huge division between the natural, non-modified, and modified organisms. So and uh, many governments uh, uh, will uh, ask of the um, require the manufacturers to label the products if they do or do not contain the genetically modified organisms. But in fact, it's uh, genetic modification is a natural process, and every time you select the larger tomato from your garden and keep it for the next year to as a seed, then you make a genetic modification. So in fact that my grandmother done that for a long time, and that is why we always eat the large tomatoes. Uh, so uh, so what she did was like she selected the largest tomato that was available this year and kept it for the as a seed for the next year, and eventually that increased the size of the tomato. And this modification is known as biotechnology, and the biotechnology is actually the use of the uh, selective breeding and to control the inheritance. And the today we are referring to biotechnology in a more narrow definition. So it's like a known as a genetic engineering, that is a modification of DNA that is optimized to produce specific characteristics. And genetic engineering makes a new strains of organisms, and this usually does not require several years for development, uh, unlike the traditional breeding. And also it allows interbreeding of different species. For example, we can interbreed the cherry and tomato, so tomato will be sweeter, or like you will have more tomatoes. So actually, uh, it's easier to combine the properties of the different plants and beneficial characteristics. That may also be the resistance to the microorganisms and diseases, so that decreases the requirements in the insecticides and can be beneficial for the consumers as well, because insecticides usually are harmful for the humans. And the recombinant DNA technology is uh, a field of the uh, botany that uh, it started with the study of the bacteriophages that and the use using bacteriophages are the types of the viruses, and the uh, there are these methods were developed during the last century, and then nowadays recombinant DNA technology can be easily used for the isolating genes from the one organism and putting them to the another organism. So this recombinant DNA technology uh, can be used for the plants and animals as well, and this allows the expression of the foreign DNA in the uh, in the body, and the that will produce the new proteins, and that will lead to the specific effects. So like here, for example, we can see the product of the genetic engineering that is uh, used. So this the gene of the bacteria is incorporated into the cotton, and then that this so on the left you can see that this cotton have produces insecticides that is usually produced with the bacteria, but now when we put the gene of the bacteria to the cotton, it produces insecticides itself, and that leads to the less uh, infestation, and that is why this genetically modified cotton is larger. So on the right you can see normal, non-modified, and here we can see the leaves are infested, and that is why the cotton is much smaller, and so that is why like the quality and the cost of this cotton is so quality is much higher and cost will be much lower, because for this you have to spend money for the insecticides, and for the people to and and the vehicles to spread the insecticides requires much energy, so then you burn fuel for that, so and that fuel will also contaminate the air, so etc etc. That is why actually genetic modification is not only harmful but also beneficial for the ecology and for the humanity. So and um, uh, and uh, there are natural mechanisms for the exchanging of the genes, and that usually can be seen in bacteria uh, with the use of the plasmids. For example, Staphylococcus aureus is producing plasmids that can transfer the resistance to antibiotics. In the same way, the other uh, microorganisms can transfer genetic material to exchange the DNA. Uh, so this is a natural genetic exchange. So and usually it occurs between the same species, like or relative species, like between bacteria and bacteria, or the virus. And but this exchange, natural exchange, is usually random and usually is not beneficial for the human. So, and in fact, it is non-beneficial in case of the resistance to the antibiotics. And to uh, to put a foreign gene into a cell requires several steps. And the gene that is selected for the uh, inserting to a different cell must be first isolated from the original cell, and then should be used for the producing a DNA molecule, that is a recombinant DNA molecule, and that should be transferred to a new cell, and new cell should identify it and express in that cell. So every of these steps requires special enzymes, and uh, so first step is the isolation of the gene of the interest, and uh, to isolate the genes we should break the original DNA into the small fragments using the restriction enzymes. So these are special bacterial enzymes that uh, have specific DNA sequences. And these restriction enzymes recognize DNA sequence and cuts the DNA whenever this sequence is recognized. So, for example, here we have this phos thymidine, cysteine, guanosine, and adenine. So this sequence is recognized and the DNA is cut at that sequence. So, and these restriction enzymes are available commercially now. So you can select the one that you are required, and then uh, 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 and this so double strand is coded at this sequence, and uh, this makes the one strand longer than the other strand. So like it should be this strand will be shorter and this strand will be longer, and and this that strand that is longer will be called a sticky end. So we will have uh, two sticky ends, and uh, so if we have two different DNAs with a sticky end that correspond, then we can connect these different DNAs to form a new DNA that will be genetically modified. And these ends then can be spliced using enzymes, special enzymes, late. So after the restriction enzymes will divide the DNA to fragments. So the next step is the combining of DNA fragments with some DNA that is a vector or the source DNA. Uh, and this vector DNA is transferring the gene from the another organism. So and usually that vector DNA is a bacteria. So uh, bacteria usually have uh, plasmids. So so bacterial DNA is usually a circular DNA. So this is a main bacterial DNA, and this is a smaller DNA that is a plasmid that is usually used by the bacteria to exchange with the genetic material with another bacteria. For example, the most known is the uh, antibiotic resistance, but also there can be resistance to the other chemicals. Uh, Other toxin producing plasmids can be also exchanged. So these plasmids are naturally used for the bacteria, but we can also use them for the carrying several genes. And uh, and when the bacteria is divided, the plasmids are also replicated, uh, and so we will have a copy of this plasmid in daughter cells. So that is why we can easily produce a large amount of the plasmids. Uh, so and when we insert the genes into the plasmids and then incorporate these plasmids into the bacterial cell, and then this bacterial cell will divide and multiply the number of the plasmids, so we can get much plasmids. Uh, so we can this way we can have as many plasmids as we want, because bacteria usually divide very fast. But some of them, for example, Escherichia coli divides every 1 second. So twice, like the number of Escherichia coli increases twice every second. So uh, and the plasmids are treated uh, uh, with the restriction enzymes, and uh, so we will take the plasmid from the bacteria, and after the treating with the restriction enzymes, the plasmid will be linear, and then we can get a DNA from the specific organism and treat it with the restriction enzyme as well, and then combine all of them and splice them with the DNA ligase, and then we will get a vector plasmid. Uh, and this is the donor, uh, so we will get a DNA from the donor organism, and now we will have a plasmid with the donor DNA. Uh, and And then we have to identify the plasmid that has a specific gene we need. And so uh, uh, to identify the plasmid of interest, we have to we have to make an amplification. So amplification is made for the bacteria that is divided. So plasmid is put to the bacteria bag, then bacteria is divided, and then we can see which of them have a which solution have a gene of interest. So then we'll select this solution. So like all bacteria will be divided to different places, and then they will multiply in these places, and then at the end of multiplication we can identify which of them is desired has a desired gene. Not all of them will be with the desired gene, but at least one of them will be. Uh, then bacteria usually don't consume plasmids, but to take that to encourage bacteria to consume plasmid, they treat the bacteria with calcium, and then uh, so this will be transferred bacteria because they have a modified uh, plasmids. Uh, uh, so now we have a mixture of bacteria. Some of them have the modified plasmid, some of them will have the normal plasmid. So and those bacteria that will have a modified plasmid will be resistant to antibiotics. So we treat the bacteria with the antibiotics, for example, penicillin, and then those bacteria that don't have modified plasmid will die, and then we will only have remaining modified uh, plasmid bacteria. So like uh, so like here we can see that uh, these plasmids uh, carry the resistant gene for the antibiotic. So and the bacteria is placed to the petri dish, and this petri dish has the antibiotic uh, agar-agar with antibiotic, and so we can see that there is no growth here because there is no plasmids, and here we have resistant plasmid. That is why uh, this colonia is growing. Uh, uh, and then the um, after the uh, after that the uh, single gem so the single gene should be identified. So uh, the to identify a gene, the genetic probe is used. and this is a radioactive labeled RNA or single-stranded DNA that is complementary to the gene of interest. And so if we want to identify a specific gene, so we know the acid sequence of this gene, and then we can synthesize a matrix RNA and this matrix RNA can be used as a probe to attach to the DNA that contains this sequence. And by the radioactive label, we can detect that under the X-ray detector, and the colony of interest can be identified by the X-ray. So, and after that, uh, the gene should be also associated with the regulatory genes that regulate uh, the turning on and off of the genes, and these genes are added to the plasmids, and so the bacterial cells can transcribe and translate the genes of interest. And then after if everything is ready, recombinant plasmid is spliced into the DNA, then the DNA is multiplied, so this is called a cloning. And then to incorporate the gene of interest to the plant, usually bacterium is used that is produces tumors of a plant. So there is a gall bacterium that is causes disease by inserting DNA from plasmid into the plant cell, and this plasmid DNA is integrated in the plant chromosome. The new inserted DNA will produce hormones that leads to the division of the plants and forming of a tumor of a plant, same as in the humans. So the and what these engineers do, they induce this tumor formation to incorporate desirable genes into the plasmid of this bacteria, and instead of the producing tumor, then the bacteria will produce it will insert the genes that are of particular interest, and these genes are placed in the same way as transmitted to the plant as the tumor, and then they can also be transferred to the next generation by sexual reproduction or asexual reproduction. Uh, Unfortunately, with this bacteria, only eudicores can be used because monocores are not susceptible, uh, and that is why we cannot use that in corn and wheat, so rice. So that is why we cannot make a rice and corn that is resistant to insects, so we have to still use insecticides for them. And the other way to introduce uh, the gene is using viruses, and they are used as a vectors, and also for the human as well, not only for the plants. Uh, and usually these viruses are treated in specific way, so they don't lead to the disease, and they just incorporate to the chromosomes. Then a biological vector also can be used, that is a plasmid or virus, and sometimes chemical modification can be also used by electric shock uh, to increase the permeability of the membrane to the DNA molecules. So not all the plants will be susceptible to these methods. So and then uh, um, it's not easy to transfer the genetic material to some species, for example, to the rice. Uh, um, and after the transfer of the gene, the culture of the plant should be regenerate to the whole plant, and this tissue technique will not work to all plants. So and not always the tissue culture can be done with all the plants. But most, like many plants, for many plants, it's available. And sometimes modified the genes for the modification are located in a chloroplast, and that is also a problem with the modification, because to increase the crops, to increase the number of organic substances, for example, protein content of a rice or a wheat, we have to modify chloroplast uh, that have their own genetic material, and uh, it's actually not available at the moment. So because uh, chloroplast will not can cannot be easily modified. So and this new research field, like determining the genomics and the DNA sequencing, is done with the computers, and is the automatic process, and usually uses decode many of the millions of amino acids, sorry, millions of the bases, and this uh, genetic sequencing is a new method that is like has been uh, opened like recently in 1990s, and uh, also recently the human genome was uh, completely identified, like I think 10 years ago, and uh, no, not 10 years, it's 15, almost 15 years ago. Uh, and also for many species, the genes are also identified, and many important crops like wheat, rice are also sequenced with the DNA, and that helps for the research and for the uh, modification of the genes. Uh, and the new uh, science, genomics, appeared, that is part of the biology that studies the DNA sequence of an organism to identify, sorry, the DNA and proteins of the different species, and also their regulation. Sorry, um, and that is very important for the application of these genes. Uh, and Usually, researchers are interested in those sequences that will code the proteins, and these DNA sequences are called expressed sequence tags. So, and uh, to to study them, usually the researchers will silence these genes and see if that will lead to the absence of the protein. Uh, and uh, and that is called a RNA interference. RNA interference is caused by the small RNA that interfere with the expression of the genes and will act on the transcription of the DNA. Uh, then we can this using this method we can study the function of the gene of the particular uh, sequence, and then after the gene is silenced, we can see the changes in the phenotype, and that will be seen by the missing the protein of interest. Then uh, because of the uh, identification of genes and sequences is very uh, information based and requires computers. A new field is appeared that is called bioinformatics, so that studies the biological calculations, so that include the DNA sequences in different species, and uh, usually it uses software and powerful supercomputers to analyze uh, data that is generated by the uh, DNA sequencing, and uh, these all are stored in databases of DNA sequences and can be compared with the functions, and evolutionary rela relationships can be found. And also pharmacogenetics uh, have, have been appeared recently. That is a new science that is based on the genes, and is the goal of this science is to customize drugs to match the patient's genes, but uh, usually this is not much helpful unless some specific cancer drugs when the pharmacokinetics is important, uh, and the uh, and usually the application is limited. However, there are many services that offer the genetic uh, genetic identification before the administration of the drugs. So uh, like many physicians, physicians now can actually ask for the genetic analysis before the or genetic screening test before the administration of the drugs that can the efficacy of which can depend on the genetic on the genes, but there are very limited number of drugs of this kind. Okay, then uh, uh, the DNA technology is also important in the medicine and pharmacology, uh, and uh, the first the first genetically modified protein uh, that is was produced for the humans by bacteria was insulin that is produced by the Escherichia coli. So that is human insulin replaced the animal insulin that was more that has many side effects, including the allergic reactions. And nowadays also the modified genetically modified insulin that is shorter acting and longer acting is also available commercially, and this is used for the treatment of the type 1 diabetes uh, uh, that allows better management of the disease with the less adverse effects. Uh, so, and this is very important step in the development of the drugs. So, and the company that first introduced this method was LA Lilly, is one of the leading companies in pharmaceuticals. Then uh, the human growth hormone is also uh, was genetically engineered and available now for the children with the growth hormone deficit uh, that will and that enables the treatment of the pituitary dwarfism, uh, and also new drugs appearing every day, including the vaccines and also the other recombinant vaccines, including the COVID vaccine as well. So these all are done with the genetic engineering. And also for the most of the antiviral vaccines like influenza, hepatitis B, poliomyelitis, and many others. And also the plants that including the potatoes, bananas, and other large uh, large fruits and vegetables are also genetically modified, uh, and uh, uh, so also DNA is used for the many other applications, uh, as well as, for example, DNA fingerprinting, and can be used for the uh, forensic purposes, like for example for investigating crime scenes. So uh, if if murder will leave some piece of the tissue, or like skin or uh, the saliva, so it can be easily identified by DNA fingerprinting. And then also treating the cancer, studying the uh, biology, and also uh, the identifying the parents of the child. So like uh, that is usually done for the identifying the father of the child. So the for example in Russia, uh, the court will require a man uh, to um, to agree for the genetic test, uh, to to be to be able to identify if uh, he was a father of the child or not. So to and whether or not he have to pay an aliments. So uh, most of the men don't want to pay because uh, actually uh, if they don't love the mother of the child, so and or were not aware that the mother, so when they have a sexual intercourse, were not aware of the mother is not using a contraceptives. So that is why uh, to prevent this, always talk with your partner to make sure you use a reliable method of contraception. Okay, so that is all for today, and thank you for your attention. And if you have any questions or comments, so then please uh, feel free to leave them below.